Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International GCSE Biology Paper 1BR for May 2023. Let's begin with the first question. Question 1 says, Lactobacillus is a type of bacterium used to make yogurt. The diagram shows a single Lactobacillus bacterium. So this is the bacterium we're talking about. We have the ribosomes, the DNA, we have the cell membrane, the cytoplasm, as well as the cell wall. And then they say, what is the function of ribosomes? Ribosomes are the site for protein synthesis, so this should be the answer. Respiration takes place in the mitochondria and photosynthesis takes place in the chloroplast, so the answer should be B. Which row in the table gives the components that are found in all bacteria and in all eukaryotic cells. So here they see the cytoplasm, of course, is going to be found in all. The cell surface membrane is found in all, but the cell wall is not found in all. Some eukaryotic cells, like plant cells, do have a cell wall. This is not found in all, and the answer should be a B. Next, they say lactobacillus cells do not burst when placed in distilled water. Red blood cells burst when placed in distilled water. Of course, this is because of possession of a cell wall. So the bacteria do have a cell wall, and these ones have no cell wall. So they excessively take in water by osmosis, increase in size, and burst. While those that do have a cell wall, when they take in water, the cell wall prevents excess expansion, and therefore they do not burst. So they say, explain why lactobacillus cells do not burst, but red blood cells do burst. This is because, like I said already, lactobacillus cells do have cell walls, but red blood cells do not have. So when placed in distilled water, water is going to enter both cells by osmosis from a higher water potential to a lower water potential. But due to the presence of the cell wall in the lactobacillus, excess expansion is going to be prevented. The cells are going to become turgid, but they're not going to expand too much, leading to their bursting. While those of the red blood cells have no cell wall, they're going to expand excessively, and they will end up bursting. In part B, describe how lactobacillus bacteria produce yogurt from milk. Milk contains lactose, and this lactose is going to be digested by the lactobacillus bacteria or used in respiration. And lactic acid is going to be produced because this is going to be an anaerobic process, anaerobic respiration. So the produced lactic acid is going to decrease the pH of the mixture which is created, and this is going to lead to the coagulation of the milk as yogurt forms. So this brings us to the end of question 1. Let's continue to question 2. Question 2, the photograph shows some seeds called lentils, which are these ones here. They say lentils are a good source of protein and are often eaten as part of a balanced diet. Describe how lentils are transported from the mouth to the stomach after being eaten. So this food is going to go through the esophagus by peristalsis, and these are rhythmic muscle contractions that push the bolus, which is the food from the mouth, towards the stomach. In part B, the recommended daily amount, RDA, of a nutrient is the mass of that nutrient required by an individual each day. The table shows some nutrients found in lentils. It also shows the percentage of each RDA for 16-year-old humans provided by 50 grams of lentils. So if somebody consumes 50 grams of lentils, this is what they're going to have. There is fat, we can see 1%, carbohydrate 11%, protein 22%, vitamin C 2%, and calcium 4%. Now we know fat can be used in respiration. Carbohydrates are a good source of sugars that are used for respiration. Proteins are used for growth and repair. Vitamin C is useful in connective tissues, and then calcium is useful in bones and teeth. So they say give one component of a balanced diet that is not shown in the table. Of course, fiber, water, and some minerals like iron and so on. Then they say lentils do not contain large amounts of vitamin C and calcium. State the long-term effect of a dietary shortage of vitamin C and of calcium. If there is lack of vitamin C, the connective tissues are not going to form well, and the person is going to get a condition called scurvy, meaning sometimes there is bleeding of gums. And if there is insufficient calcium, of course, they could get rickets because their bones are going to be weak. The next part says, calculate the mass in grams of lentils that a 16-year-old needs to eat each day to provide their RDA of protein. They want you to give your answer to two significant figures. From the table, 22% of what they need is going to be provided by 50 grams of lentils, meaning 100% is going to be provided by 50 over 22 times 100, giving us 230 grams if you write to two significant figures. The next they say describe how protein is digested in the human alimentary canal. This is easy. Digestion occurs in the stomach, in the duodenum, as well as the small intestine. In the stomach, proteins are going to be broken down into peptides using pepsin enzyme. The conditions are going to be acidic, provided by HCl. And then in the duodenum, the pancreatic enzyme trypsin is going to break down the proteins into peptides. And then in the small intestine, the peptides are going to be broken down into amino acids with the help of peptidases. So this brings us to the end of question 2. Let's continue to question 3. Question 3, the diagram shows a forest food web. So this is the food web, and we can see there are many components and there are many food chains. They say how many secondary consumers are there in this food web. To be able to answer this question, we have to look at the possible food chains. So I have this food chain here. This is a producer, primary consumer, and this is a secondary consumer. If I consider from here through that to that, again, producer, primary consumer, secondary consumer. Here we have producer, primary consumer, secondary consumer. So the fox is one example of a secondary consumer. And then we have one, which is a producer, primary consumer, secondary consumer. And then, so this one is also a secondary consumer in that specific food chain. Then we have this one here, producer, primary consumer, secondary consumer. And we have producer, primary consumer, secondary consumer. So we have 1, 2, and 3 secondary consumers. So my answer here was a 3. The next they say which term describes all the organisms in the food web together in their environment. This is going to be an ecosystem. This is organisms interacting with each other as well as with their environment or with their surrounding. So that is going to be an ecosystem. The next part says the fox receives 1% of the energy present in the oak tree. The fox receives 0.04% of the energy present in the grass. Explain why a higher proportion of the energy reaches the fox from the oak tree than reaches the fox from the grass. So I placed this here so that we can be able to see. If we look at this diagram here, we can see the oak tree does not have so many primary consumers. I'm talking about food chains connected to it. We have one food chain, and that's the second. While from the grass, we have that food chain. There is another and another. Then also this one here. From the grass food chains, the fox is either a secondary consumer or it's going to be a tertiary consumer. While in this case, when we go to the oak tree, the maximum the fox can be is a secondary consumer, and that means it's going to be lower in the chain, so it's going to be able to access more of the energy present within the oak tree in comparison to what we see in the grass. So I said in the oak tree food chains, the fox is a secondary consumer, while in the grass food chain, the fox is a tertiary consumer. Energy losses occur at each trophic level, so less is transferred per level, meaning from the previous one to the next. And losses can be due to respiration. They could be due to uneaten parts like roots and stems. They could be due to undigested parts or excreted parts, and so on, and others could die before they are consumed. In part B, a student uses this method to estimate the total area of the woodland floor that is covered by grass. Randomly place a 0.25 meter squared quadrat in one location of the woodland. Then estimate the percentage of the quadrat that is covered by the grass. Repeat at one other location. Calculate the mean percentage covered by grass for both quadrats and measure the total area of the woodland floor. Then calculate the total area of the woodland covered by the grass. They say state why the student placed a quadrat randomly at each location. Placing the quadrat randomly prevents being biased during sampling. In the next part, they say explain how the student could improve their method to obtain a more reliable estimate of the total area of the woodland covered by grass. Based on the information given, these only sampled in 2 areas, so you could repeat with more throws and then calculate the mean. And afterwards, they could identify the results that deviate far from the mean, which are the anomalous results, and then those could be eliminated. The diagram shows the dimensions of the woodland. So the dimensions are 100 here, we have 50, we have 80, and that means this is going to be a 20. The student finds the mean percentage of the 2 quadrats covered by grass. Use this value to calculate the total area of the woodland floor covered by grass. The woodland area is going to be area of this rectangle plus area of this triangle. So since this is 100 and that is 80, I can be able to know that this is 20. So this area here is going to be a half times 20. times 50, which is the height, as you can see here. And the area of this is going to be 50 times 80, which is what you see here. So total area of this woodland is going to be the area of the rectangle, which is 80 times 50, plus the area of the triangle, which is what we have here. And the total area was 4,500 meters squared. Now the area occupied by grass, which is 65%, is going to be 65 over 100 times the total area, and this is going to be 2925 meters squared. And that was my answer for question 3. So this brings us to the end of question 3. Let's continue to question 4. Question 4. Yeast is a fungus that can respire anaerobically. What substance is the cell wall of a yeast cell made of? It's going to be made of chitin, so the answer should be B. The next part says which row of the table shows the substances produced by yeast during anaerobic respiration. Yeast produces ethanol as well as carbon dioxide, so the answer here should be A. Lactic acid is produced in animals. A student uses this apparatus to investigate the effect of temperature on anaerobic respiration by yeast. Here we can see this is the experimental setup. We have the paraffin that is going to prevent oxygen from entering the experimental setup and therefore preventing aerobic respiration from being carried out. We also have the yeast cells in the glucose solution. Of course, the glucose is there to provide an energy source or a carbohydrate that is going to be used in respiration. On the other side, we have the gas bubbles. Of course, we can see because respiration is going to be carried out, carbon dioxide is going to be produced, and that carbon dioxide passes through here to the other side. If this was lime water, then we'll see it turn milky. They say place yeast cells and glucose solution into a test tube and keep the test tube at a temperature of 25 degrees Celsius. Then cover the yeast and glucose solution with paraffin oil. Count the number of bubbles produced each minute for 20 minutes. Repeat at a temperature of 37 degrees Celsius. So remember, the number of bubbles produced within 20 minutes is going to give us the rate. So if there are more bubbles produced in 20 minutes, then it means the rate is going to be higher. Then they say the student draws a graph to show the total number of bubbles that have been produced by the end of each minute for the 20-minute period. The graph shows the student's results. So this is the graph. We have the total number of bubbles produced on the vertical axis, and then we have the time in minutes. And we can see from the key, this is carried out at 25 degrees Celsius, and this is carried out at 37 degrees Celsius. This has a higher rate because the line is steeper in comparison to this one. However, by the end of the experiment, the total number of bubbles produced were the same, although this produced those bubbles within a shorter period of time, which is just about 10 minutes, while these had to reach about 18 minutes in order to produce those. So this one had a higher rate in comparison to that, meaning the higher the temperature, the higher the rate of reaction, because there is more kinetic energy that is causing more collisions to occur per time. So here they say state the function of the paraffin oil. Paraffin oil, like I already said, it prevents air containing oxygen from entering the test tube, and this is going to ensure that anaerobic respiration is going to be attained. The next they say describe a method the student could use to keep the temperature for the yeast and glucose mixture constant. They could use a water bath or a thermostatically controlled water bath to ensure that the temperature does not deviate or does not change. The next they say calculate the rate of bubble production from 0 minutes to 8 minutes when the yeast is at 37 degrees Celsius. To do this, I'm going to take you back to the graph. Because this is a line, just get 2 points that you can accurately determine. For example, I use this point, which is 2.510, and this one here, which is 728. So I'm going to do change in y, which is 28 minus 10 divided by 7 minus 2.5. So this is what I did here, and I got 18 divided by 4.5, which gave me 4 bubbles per minute, and that was my rate. Then down here they say explain the change in the total number of bubbles produced from 0 to 20 minutes at 25 degrees Celsius. From this graph at 25 degrees Celsius, we can see the rate increases until about 18 minutes, and then there is no change in the rate. And this is because the rate of reaction is stopped. It could be due to production of waste that is affecting the rate. For example, carbon dioxide is produced in this reaction. It could be that the accumulation of CO2 is changing the pH and causing the enzyme-controlled reactions not to occur properly. It could be because the nutrients are finished, and therefore the rate is going to be hindered by some other factor other than temperature. So here I said initially the number of bubbles increase per time until 16 minutes. The rate decreases between 16 and 18 minutes and becomes constant at 18 minutes. The amount of glucose could be a limiting factor. It can also be due to excess ethanol production that destroys the yeast. Next, they say explain the difference in the rate of bubble production between 0 to 10 minutes at 37 compared to 25 degrees. At 37, there was a higher rate in comparison to 25. So I said at 37, there is more kinetic energy than at 25 degrees Celsius. So more successful collisions occur between the particles involved per time. So the rate is higher and more bubbles are produced. These are the bubbles of carbon dioxide gas. Also at 37 degrees Celsius, due to a faster rate, glucose runs out faster and the rate levels off at 10 minutes as the amount of glucose limits the rate. You could also say that carbon dioxide has been produced, changing the pH and therefore causing the rate of reaction to slow down or to stop. Next, they say describe how the student could modify the experiment to give more accurate results. Now, if we go back here, they counted the bubbles of gas produced in this experiment. And of course, this is not going to be efficient since different bubbles could have different volume of gas or some gas could dissolve in the liquid. So it's better to accurately collect the volume of gas produced, and here using a syringe would be a better way. So I say the student could use a syringe to collect the volume of gas produced instead of counting bubbles, since different bubbles can have different size or they could contain different volumes of gas, which could be misleading in the results. So this brings us to the end of question 4. Let's continue to question 5. Question 5. The diagram shows the structure of a human heart with 2 chambers labeled W and Z and 2 blood vessels labeled X and Y. So this is the heart. We can see this is the pulmonary artery. That is going to be the aorta. This is going to be the right atrium, and this is the left ventricle. So they say which row of the table correctly identifies chambers W and Z. Of course, like I've already said, it's going to be C. W is the right atrium, which we see here, and this is going to be the left ventricle. The next they say give 2 differences between the composition of blood in X and Y. And again, here they're asking for composition of blood. It means we're going to be talking about oxygen and carbon dioxide concentration. Y contains blood with greater oxygen concentration than X. Of course, this is blood going to the rest of the body through the aorta, so it has greater oxygen concentration and it has lower carbon dioxide concentration. So my second answer was Y transports blood. This should be lower. So Y transports blood at lower carbon dioxide concentration than blood transported through X. Next, they say a scientist investigates the effect of exercise on the heart rate of 2 people. So they are carrying out on 2 people. One person is a trained athlete and the other is an untrained volunteer. The heart rates of both individuals are measured at rest, meaning at 0 minutes, and both individuals then exercise for 6 minutes and then rest for another 6 minutes. The table shows the heart rates of the untrained volunteer and the trained athlete at rest, during exercise, and after exercise. So here they say the cardiac output is the volume of blood pumped out by the left ventricle in 1 minute, and the stroke volume is the volume of blood pumped out by the left ventricle in 1 beat. The cardiac output of the resting human is 4,900 centimeters cubed per minute. They want you to calculate the stroke volume of the trained athlete when at rest, when their cardiac output is 4,900 centimeters cubed per minute. They want you to use this formula. Cardiac output is equal to stroke volume times heart rate. So in order to calculate stroke volume, I have to make this the subject. Stroke volume is going to be cardiac output divided by heart rate. But since I'm given this as my cardiac output, which is 4,900 centimeters cubed per minute, and I have my heart rate for this person, which is going to be 55 beats per minute, or BPM, my answer is in 3 significant figures, so I got 89.1 centimeters cubed. The next they say, plot a graph to show the heart rate of the untrained volunteer and the trained athlete when resting and during exercise, from 0 minutes when at rest to 12 minutes. Use a ruler to join the points with straight lines. Now, I really tried hard to find a suitable scale, but it was really going to be complex, so the vertical axis is not from 0, and again, I began at 50 because the scale was going to be very complex. So with my graph on the vertical axis, I have heart rate in beats per minute, and then on the horizontal axis, we have time in minutes. So these are the points that I connected. This is quite easy based on what you have from the table, and then just connect the points. I have my key here. This one here, the part in blue is for the untrained volunteer and the part in red is for the trained athlete. So this is quite easy. You just have to make sure that your graph can fit at least half of the space provided, and then you'll be fine. And next they say, explain why the trained athlete can run faster than the untrained volunteer. They want you to use the information in the table to support your answer. If you go back to the table, we have information given. Of course, there is the resting heart rate. The trained athlete has a lower resting heart rate in comparison to the untrained volunteer. And because this athlete trains, he or she is used to having bigger volumes of blood pumped through their heart for specific periods of time. So I could say they could have a larger heart. So I say a trained athlete has a larger heart than an untrained athlete. So the larger heart pumps large volumes of oxygenated blood with glucose to the muscles. This ensures rapid production of ATP for muscle activity, which is going to be muscle contraction. And also this ensures that lactic acid from anaerobic respiration can be quickly transported to the liver for its metabolism in order to overcome oxygen debt. That's why after exercise, the heart rate of the trained athlete returns faster to their resting heart rate in comparison to the untrained volunteer. So this brings us to the end of question 5. Let's continue to question 6. Question 6. Variegated leaves have areas that are green and areas that are white. A student uses this method to investigate the effect of light on photosynthesis in a variegated leaf. So they place a plant in the dark for 24 hours. This is to discharge the plant. And then they wrap a strip of black paper across a leaf. They shine light on the plant for 24 hours. This is to ensure that photosynthesis takes place. But putting a black strip we saw here was to ensure that in those areas that are sealed, no sunlight is exposed and therefore there will be no photosynthesis. So they say remove the black paper and use iodine solution to test the leaf for starch. So the diagram shows the apparatus and the student setup. So you can see this is the lamp providing the light. This is the place where the black paper has been placed so that light does not shine in that area and therefore there will be no photosynthesis. And after the experiment, we expect no starch to be present there. The first question says complete the balanced chemical symbol equation for photosynthesis. This is going to be 6 carbon dioxide plus 6 water gives us glucose and 6 molecules of oxygen. Then in part B, they say state why the plant was placed in the dark for 24 hours. This is to discharge the leaves. The next part says diagram 1 shows the position of the black paper on the leaf. So we can see the white areas and then the black areas and then the green areas, of course. We expect these white areas not to turn blue-black because there is no chlorophyll. And then the black area, of course, is not going to turn blue-black because there was no light exposed to that area. So there will be no starch present because photosynthesis was not carried out. And in the green areas, because they were exposed to light, we will see them turn blue-black because chlorophyll was present and also photosynthesis took place. Complete diagram 2 to show where the variegated leaf would appear black after testing with iodine solution. Like I said already, this region and this region, number 1 is green, so it has chlorophyll, and number 2, it was exposed to light, so photosynthesis took place, so it's going to appear blue-black when you test with iodine, whereas this region that was wrapped with a black paper is not going to photosynthesize, and these regions here are white, so they do not have chlorophyll, so there will be no photosynthesis. In part C, the student observes that the leaves on different ivy plants seem to be different sizes depending on the amount of sunlight the plant receives. They want you to design an investigation to test whether the amount of sunlight received by ivy plants affects the size of their leaves. They want you to include experimental details in your answer and write in full sentences. In this experiment, the amount of sunlight is going to be the independent variable, so we have to find ways of exposing the ivy plants to different light intensities. So I said set up experiments with ivy plants at different light intensities. And since we're using plants, they should be the same species, they should be the same age. Of course, you could also use the same plant. Now, at every light intensity, we have to repeat the experiment and calculate the mean. Then we have to let the plants to be exposed to each light intensity for the same period of time. It could be about 5 days. Then we measure the length of the leaves from each light intensity. And again, we have to measure both starting and final lengths. You want to be able to calculate the change. So measure the starting leaf size or leaf length, and then the final leaf length, and then calculate the percentage change in leaf length at each light intensity to determine the best light intensity. While doing this experiment, we have to keep some variables constant, so we could keep temperature, humidity, minerals, water, carbon dioxide concentration, the fertilizers of the soil, the pH of the soil. All these have to be kept the same in all experiments or in all experiments at all light intensities to ensure that the only thing causing the changes or differences in the results is due to the variation of the independent variable and nothing else. So this brings us to the end of question 6. Let's continue to question 7. In question 7, the diagram shows the human female reproductive system, which is what we see here. In part A, they say, where does fertilization normally occur? Fertilization is going to occur in region A, which are the oviducts, or some people say the fallopian tube. The next they say, state how estrogen and progesterone affect structure C during the menstrual cycle. Structure C, that is where we have the uterus. So estrogen is going to cause the thickening as well as the repair of the uterine lining. And then progesterone is going to maintain whatever has been thickened to ensure that it prepares for pregnancy. In part B, in vitro fertilization, IVF is used to help some people have children. Eggs are mixed with sperm in a lab or in the laboratory, and embryos are then transferred into a uterus. They want you to describe how the embryo forms after the egg and sperm have been mixed together. After mixing, both the egg and sperm nuclei fuse together to form a zygote, and then the zygote goes through mitotic division to produce more cells that make up the embryo. Actually, I could go further to say cell differentiation takes place, because in the embryo we have various cells that have differentiated, so cell differentiation takes place so that the produced cells are going to be specialized to become the different cells within the embryo. And again, remember, the embryo has various cell types, but all these cell types came from one zygote, so there should have been differentiation in order to produce these many cells that are specialized to perform different functions. Then here they say if more than one embryo is transferred to a uterus, IVF can result in multiple births, such as twins or triplets. Pregnancies that produce more than one baby can be a health risk. In the United Kingdom, since 2007, the recommendations for the number of embryos transferred are: a person under 40 should have only one embryo placed into their uterus, and the person over 40 may have 2 embryos placed in their uterus. This is because for people over 40, of course, the chances of getting pregnant are decreased, so it's allowed to put more embryos, but those who are below 40, the chances are kind of still higher, so they recommend to put only one embryo. So they say the graph shows the percentage of IVF treatments that lead to multiple births from 1993 to 2019. So from the graph on the vertical axis, we have the percentage of IVF treatments that led to multiple births, and on the horizontal axis, we have the time in years, of course. And overall, we can see as time increases or as time passes, there is a decrease in the percentage of IVF treatments that lead to or that led to multiple births. Then down here, they say the table shows the success rates of IVF in the United Kingdom in 2019 for people of different age groups. Those under 35, the success rate was 32%. Those between 35 to 37, 28%. 38 to 39, 19%. All the way until 45 to 50, which is just 4%. So we can see that as people age, the percentage of IVF treatments being successful decreases. So here they say discuss the effects of limiting the number of embryos transferred, and use the information in the table and the graph to support your answer. The percentage of IVF treatments that led to multiple births decreased, and of course this could be due to the regulations we saw here. So here I said before 2007, the percentage of multiple births generally decreased, but with occasional oscillations. But since 2007, the percentage of treatment leading to multiple births has steadily decreased. The steady decrease could be because of implanting fewer embryos or increase in the expenditure from IVF-related medical bills. More embryos are inserted in the uterus of women with increase in age. However, the percentage of multiple births decreases with increase in age. And this is because as women age, they have lower rates of successful implantations and, of course, less healthy pregnancies. From this information, we were not given the number of females who participated in this study. We do not know their health history because not being able to carry a healthy pregnancy is not going to be only due to age. There could be some other health risks. So we have no health history, or there is lack of sufficient data like health history as well as genetics. And also the number of people used per study was not included, and this could be useful in determining reliability of the results. And finally, the research was only carried out in the United Kingdom, so similar research should be repeated with women from other global regions to make fair conclusions. So that is in my discussion part. There is benefits to this as well as things I think they could have improved. So this brings us to the end of question 7. Let's continue to question 8. Question 8, the diagram shows the carbon cycle, and this is the carbon cycle. So they say which letter in the diagram represents respiration. Respiration should be when organic compounds are converted into gaseous carbon dioxide. So I already labeled this. We can say this is organic compounds. In decomposition, of course, CO2 is produced, so this is respiration. That is respiration. And we can see organic compounds in plants becoming carbon dioxide, which is inorganic. That is respiration. So why is going to be respiration? I already labeled the other. This is death. That is death. This one here is going to be photosynthesis, as we can see there. That is going to be fossilization. And that is going to be combustion. Next, they say give the name of the process labeled W. This is photosynthesis because we can see carbon dioxide from the atmosphere is being converted into organic compounds within the plants, and that only occurs through photosynthesis. In part B, carbon dioxide, methane, and nitrous oxide are 3 greenhouse gases. The table shows a comparison of these greenhouse gases. The global warming potential is the ratio of the heat absorbed by a greenhouse gas in the atmosphere relative to the heat absorbed by the same mass of carbon dioxide gas. So from the table, we have carbon dioxide, methane, and nitrous oxide. We have the percentage of all greenhouse gas emissions. Of course, this has the greatest percentage. This has the lowest percentage. And then we have the global warming potential. This has the highest global warming potential. And the length of time the gas stays in the atmosphere, this has the greatest length. Now, this being produced in the highest concentration and yet staying in the atmosphere longer could make it a very potent greenhouse gas. And although this is produced in a very low concentration, it has greater potential for causing global warming, and it stays in the atmosphere longer than methane. Then methane is going to be intermediate, of course. So my conclusion, I could think carbon dioxide is going to be able to cause greater global warming effects in comparison to the others. The first question says name one other greenhouse gas. Of course, this is going to be water vapor. And here they say evaluate the information in the table to identify which of these gases is likely to contribute most to global warming. In my choice, I took carbon dioxide, and this is because the percentage of carbon dioxide is the highest and it takes longer to be removed from the atmosphere. So I said carbon dioxide is likely to contribute most to global warming. It has the greatest percentage of all greenhouse gases. This is mainly because of the human activity like burning fossil fuels. Also, carbon dioxide stays in the atmosphere longer than other gases. However, it has the lowest global warming potential. Then when we go to methane, it contributes 16% of all greenhouse gases, and this is lower than that of CO2 as well as methane. It stays less in the atmosphere. However, it has greater global warming potential than carbon dioxide, so they could be that this is going to be potent as well. The next is nitrous oxide, and this has the lowest percentage of greenhouse gases released. It stays in the atmosphere longer than methane. However, it has the greatest global warming potential, so that could be dangerous as well. But in the end, carbon dioxide is what I chose as the most potent one. So this brings us to the end of question 8. Let's continue to question 9. Scientists have developed transgenic crop plants that are resistant to herbicides, which are the weed killers. They say state what is meant by the term transgenic. Transgenic means having DNA from 2 different species, 2 or more different species. So this is the transfer of DNA from one species to another, so to create a transgenic organism. Next, they say suggest why growing herbicide-resistant crop plants is beneficial to farmers. If farmers are going to use herbicides, it means herbicide-resistant crops are not going to be affected. They will thrive. However, the herbicide is going to destroy the weeds only and not the crops. So this is going to decrease the competition for nutrients and thereby increasing the yield of the crops. In part B, the photograph shows a weed called Palmer amaranth that is often found in fields growing soybean. So this is Palmer amaranth. They say Palmer amaranth is a weed that has become resistant to many different herbicides. In some populations of Palmer amaranth plants, a recessive allele, which is small r, makes the plant resistant to herbicide. A dominant allele, capital R, makes the plant not resistant to herbicide. Give the possible genotype of Palmer amaranth plants that are not resistant to herbicides. If they're not resistant to herbicides and they've told us this is going to be capital R, so it's either going to be homozygous dominant, like in this case, or it's going to be heterozygous, so I wrote that and that. The next they say a Palmer amaranth plant that is resistant to herbicides, and again remember the recessive one is resistant, so if it's resistant it means it's going to be in the homozygous recessive version. So this is crossed with a Palmer amaranth plant that is heterozygous for herbicide resistance, which is that. They said draw a genetic diagram to show the genotypes of the parents, the gametes they produce, and the genotypes and the phenotypes of the offsprings. Because we are crossing that and that, the parental genotype is going to be capital R small r and small r small r. This is going to be the recessive one and therefore the one resistant to the herbicides. The gametes are going to be from here, capital R and that, while from here is going to be that. In carrying out a genetic cross, we will have that and that for this parent, and then we'll have small r from the other parent, which is that. Here I got that, which is heterozygous, homozygous recessive, heterozygous, and homozygous recessive. So the offspring genotype is going to be the ratio of 1 to 1. This is for heterozygous to homozygous recessive. And then the offspring phenotype is going to be 1 non-resistant and 1 resistant. And I'm talking about the ratio 1 to 1. Give the probability of the cross in B2 producing a plant that is resistant to herbicides. If it's going to be resistant to the herbicides, which we have as that and that, so it's going to be 2 out of the total, which is 4, giving us a probability of 1 over 2. The next they say explain how Palmer amaranth plants have evolved to become resistant to herbicides in areas where herbicides are used frequently. Every time they talk about evolution, you have to talk about a mutation occurring which causes variation, and because of variation, there could be selection pressure that causes some to survive while others die, and those that survive are going to reproduce and pass on their alleles to their offsprings. So I said a mutation occurred, and this led to variation, including a herbicide-resistant allele. On exposure to the herbicide, which is selection pressure, the plants with a resistant allele survived, and they reproduced, so the alleles were passed on to the offspring plants. In plants such as Palmer amaranth, where the allele for herbicide resistance is recessive, no non-resistant weed occur after 5 years of using the herbicide. However, in other plants, where the allele for herbicide resistance is dominant, some non-resistant weeds occur after 5 years of using the herbicide. Explain this difference in number of non-resistant weeds after 5 years of using the herbicide. Now, if the herbicide-resistant allele is recessive, of course, it's going to be expressed when it's in the homozygous recessive version, meaning those that will be resistant are going to be only the homozygous recessive ones. And so those that have the dominant allele over time are going to be destroyed or they're going to pass away. And that means over time we'll only have those that are resistant, which are going to be with the recessive or in the homozygous recessive version. So I said if the resistance allele is recessive, only homozygous recessive plants survive once exposed to the herbicide. So if the resistance allele is dominant, surviving plants can be both homozygous dominant and heterozygous, so the recessive allele can still remain in the population among the heterozygotes. And when you breed two heterozygotes, this can lead to production of a non-resistant plant, and this could be 5 years after using the herbicide. So this brings us to the end of question 9. Let's continue to question 10. Question 10. The diagram shows a structure of a human eye. So this is the human eye here. They say give the name of structure Z. The structure Z is the iris. And then they say describe how structure X and Y control the shape of the lens when focusing on a near object. X is the ciliary muscle And what is the suspensory ligament? So when focusing on a near object, remember that light rays are going to approach the eye in a diverging way, so we need to cause bending. So here the lens has to become thickened. So on focusing on a near object, the ciliary muscles have to contract, the suspensory ligaments are going to slack, the lens is going to become more convex, or I would say it's going to be thicker in order to refract or bend the light more so that it can be focused onto the retina in order for the image to be interpreted. In part B, a cataract occurs when the lens becomes cloudy. Severe cataracts are the main cause of blindness around the world. Explain why people with cataracts are unable to see clearly. If the lens is cloudy, then less light is going to enter to the eyes, and therefore the rods and cones of the retina are not going to be stimulated well to create electrical signals or electric impulses. So fewer impulses are going to be sent to the brain, and the person will not be able to see clearly. The next they say exposure to UV light is thought to be a risk factor for the development of cataracts. The table shows the results of a 25-year investigation into the effect of different mean hours of sunlight on agriculture workers in one South Asian country. So here we have the mean daily hours of exposure to sunlight. We can see they're increasing, and the number of people in the group, and the number of people developing cataracts, we can see from 2 to 35. Of course, the number of people is increasing, but we can see the percentage. I calculated is 2%, 1.9%, 2%, 1.6%, 3.2%, and 6.5%. So here they say comment on the results of this investigation. From this experiment, we can see it was carried out for 25 years, therefore a long-term study, so it can be more believable. They used more people, so the results could be reliable. So I say the number of people developing cataracts increases with exposure to sunlight. That is true. From here, we can see with more exposure to sunlight, the number of people developing cataracts increased. But of course, this is not representative of the percentage. Of course, we see the percentages are varying, or there is some oscillation. So however, different groups have different number of participants, so using percentages is a better way of interpreting the data. As shown from the table on the previous page, the percentages with increased exposure to sunlight are 2%, 1.9%, 2%, 1.6%, 3.2%, and 6.5% respectively. Between 7 to 10 hours of exposure to sunlight, there is almost no difference in the percentages. However, after 10 hours, there is increase in the percentage of people who develop cataracts with sunlight exposure. However, more information about the participants like genetics, gender, etc. should be included to make valid conclusions. And again, every study that is carried out, we may not know if it's a multifactorial condition. For example, developing cataracts could be genetic. It could be based on gender. Just using the information given in the table to make a conclusion will not be fair enough, so more information should have been provided. So this brings us to the end of question 10, as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.